Okay guys, so last time we reviewed our photosystem 2, photosystem 1, then we went over the Kelvin cycle, and if you remember, the goal of the Kelvin cycle is to use our ATP formed from photosystem 2, our NADPH formed from photosystem number 1, and use those along with carbon dioxide, which is a new character in our Kelvin cycle, in order to make glucose, okay? Now, in the end of it, we formed this molecule called PGA. Another name for that molecule is G3P, which we'll talk about in the notes. Okay, I just want to make sure you guys knew that. But anyway, steps of the Kelvin cycle. So we're going to start off by taking in carbon dioxide. And how does a plant take in carbon dioxide? Well, they don't really have mouths, but on the underside of their leaves, they have these little tiny things that look like coffee beans, and they are called stomata. And what will happen here is the stomata will open, and when they are open, carbon dioxide can come in, and the O2 that was formed by the splitting of water can actually leave as well. And that's pretty much how they are able to breathe, okay, through their stomata. So there's your first step of the Kelvin cycle, is we got to be able to get carbon dioxide in. Without carbon dioxide, we can't fix carbon with our RUBP. Second step is we are now going to, once carbon dioxide is in, we are going to fix our carbon. So the CO2 is going to combine with our 5-carbon molecule, which is ribulose biphosphate, RUBP. Okay, it is a 5-carbon molecule, and on each side of it, it has a phosphate. Once we form that 6-carbon one, it's unstable. We talked about that last time, which is why we are going to split that one. So we split it into PGA, okay? Now, for our intents and purposes, we could say PGA and G3P are pretty much the same thing. Only difference is like between the phosphates there. All right. So once we get our PGA, it's transferred into G3P, which is glyceraldehyde. Okay, that's the fancy name for it. But anyway, guys, it's still a 3-carbon sugar. It's still just like PGA. So you can use these terms, you know, you can use both of them. Whenever we are going to leave the Kelvin cycle, we are going to take 2 PGAs or 2 G3Ps, and that's how we are going to get our glucose, okay? In this process, we are going to use ATP and NADPH. Once we use our ATP and it turns into ADP, the ADP is going to go back to photosystem 2, and our NADP plus is going to go back to photosystem number 1. All right? So that's the cycle between our ATP and our ADP and our NADPH and our NADP plus. And like I said, guys, the G3P or the PGA, once it leaves, okay, it's not going to be converted into glucose right away. It goes through a bunch of different things. It's going to actually leave our chloroplast before it gets converted into sucrose or glucose or what have you, okay? So it's not an immediate process, but we will get glucose in the end, and that's our goal of the Kelvin cycle, is to get glucose. Oops. Okay, so the rest of our PGA or our G3P, whatever we have left over, which if you remember from yesterday, it was 30 carbon atoms. Those 30 carbon atoms are going to get converted back from G3P into RUBP so it can continue through the cycle once again. All right, and that's that part right there. Okay, so we're going to go over some alternative pathways to the Kelvin cycle. All right, usually we do PS2, PS1, and then go straight to the Kelvin cycle. But there are a few things that can affect how our sequence is going to go. We would like it to be right in order. It just, hey, PS2 goes, okay, now PS1 goes. All right, that's done. Let's just do the Kelvin cycle. But sometimes the Kelvin cycle cannot fire. So we talked about the stomata. They let the carbon dioxide in, and they release O2 so we can breathe the O2. When the stomata are closed, the CO2 cannot get in, and the O2 levels inside are just going to keep rising, and this is actually going to stop the Kelvin cycle. All right, so again, there's your stomata there. Now, why would we not want to open our stomata is the question. Okay, well, we need carbon dioxide. We want to let this O2 out. Just open up, all right? Well, plants that live in really, really hot areas, they don't want to open their stomata, because when they open their stomata, they will actually lose water when the stomata is open, and they need water to do the photosystems. All right, so if they can't lose water, they don't want to do that, that means they can't bring in carbon dioxide as well. And again, this is for plants that live in very hot environments, okay? So if the plants can't go through the Kelvin cycle, there is an alternative pathway called the C4 pathway. The Kelvin cycle is also called the C3 pathway because we end up with a 3-carbon molecule at the end. C4 pathway is all we're going to do is we are going to fix our carbon into a 4-carbon molecule. It's just a little different pathway. If you take my AP class, we will go over this whole pathway in depth. But for right now, all you guys have to know is there is an alternative pathway. It makes a 4-carbon molecule instead of a 3-carbon molecule. And some examples of it, corn, sugar, crabgrass, okay? Some plants like this go through the C4 pathway. All right, so there's all of your lovely, plants there. There's your corn, your sugar cane, your crabgrass down there. All right, the other one is called the CAM pathway. And this is for plants that live in really, really, really hot areas. Examples, cactus, pineapples. What they're going to do is they are still going to want to go over the sequence with PS2, PS1, and then the Kelvin cycle. However, the issue here is that they cannot open their stomata during the day. So what they're going to do is they're going to save the products of PS2 and PS1. They're going to save that ATP. They're going to save that NADPH. And then at night, when they are not worried about losing water when they open up their stomata, they'll open their stomata up, they'll take that carbon dioxide in, they'll let the O2 out, and when they do this, then they can go through the Kelvin cycle. Then they can use the ATP, then they can use the NADPH and go through that Kelvin cycle to make our glucose. So that's CAM pathway. All they do, the only difference is they're going to do the stomata portion where they need the carbon dioxide. They're going to do the Kelvin cycle at night and do the photosystems during the day. Okay? All right, so things that can affect photosynthesis is light intensity. As our light increases, we are going to have an increase. Here, we'll just say, I'm just going to call it P rate for photosynthesis rate. Just going to make a little graph here. This is going to be time. Okay. So as our light, I'm sorry. That should not have been time. My fault, guys. Okay. This should be our light intensity. Okay. Obviously, as we go from left to right, the light is getting more intense. So what's going to happen here is our light increases, our photosynthesis rate, we are going to be able to make more and more and more and more of our glucose. However, there is a point where if we keep increasing the light intensity, it's actually not going to help us anymore. Okay? It's not just going to keep going up. It's actually going to plateau on the top. Why is it going to do that? Well, all the electrons have become excited already. There's no more that can become excited to increase photosynthesis. So once we get so much light in there, we are going to increase photosynthesis originally, but then it's going to stop. It's going to plateau across, okay? So that is light intensity. All right. So, like I said, just in the graph, there is a point where all the electrons have become excited and we can't increase photosynthesis anymore, all right? obviously we'd have a higher photosynthesis rate here than here. But in this area, there is a point where the plants cannot photosynthesize any faster. It's like everyone is working as hard as they can. No one can go any faster than they're already going. All right. Carbon dioxide levels. It is the exact same graph here pretty much. So we'll have CO2 levels, and we'll have our photosynthesis, our P rate over here. And it's the exact same thing. As we increase our carbon dioxide, it's going to go higher, 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 and then it's going to plateau once again, because it doesn't matter how much carbon dioxide we bring in, we can't work any faster than we're working, okay? So that is your carbon dioxide levels. Once we get them so high, our photosynthesis rate cannot increase. And then our last one is going to be temperature. Now, temperature is a little weird. So I'm actually going to put some numbers in this graph. And given we are in Pennsylvania, I will use plants that are kind of like the ones we would see with temperature around here. We'll say this is 0. Say this is, I don't know, 50. Say this is 100. Oh, we'll go to 150. Why not? And that's degrees Fahrenheit. Okay, so at 0 degrees, yeah, the plant is pretty much frozen. The water's frozen, everything's frozen. It is not a good time. So our photosynthesis rate, let me do a different color here. Our photosynthesis rate is going to be pretty much none, maybe a little bit, I don't know, somewhere around there. Now, once we get above freezing, our photosynthesis rate is going to increase, 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 increase. But then we're like around 80, 90, somewhere around there, getting really hot, okay? Maybe even around 100. Well, our photosynthesis rate is increasing, but then what's going to happen is it's going to decrease and decrease and decrease, and then slowly get to pretty much 0. Why? Well, what's happening here, guys, is we need 3 things for photosynthesis. We need water, okay? Water's not a big deal. Okay, we can keep the water in. We can keep it all in this very large central vacuole the plants have, all right? Sunlight, if it's over 100 degrees, we definitely got some sunlight. Problem is the carbon dioxide. If it is too hot, the plants are not going to open up their stomata. So if the plants don't open up their stomata, they're not going to get any carbon dioxide. No carbon dioxide, no glucose can be formed. So that's why the photosynthesis rate starts dropping at very high temperatures. All right, here's your bullet on it. There you go. So past a certain temperature, the stomata will close, and this will limit the amount of carbon dioxide that the plant will have to carry out photosynthesis. Okay, they did a little study. Now this is in degrees Celsius. Don't think this is in Fahrenheit, but they found that around 20 to 25 degrees, the plants actually grow really well. If it gets too hot, they're not going to grow as much. They're not going to make as much glucose. They're not going to have as much ATP from cellular respiration. So they're not going to grow as quickly. All right, guys. So that is the end of chapter 6. And you guys can start studying for your test. And I will see you later.